Good day everyone, especially to our subject teacher, Miss Marisa Banuelos. My name is Queenie Joy of Friel.
My name is Janine M. Laguna.
My name is Hazel Dulalia.
My name is Crisey M. San Juan.
My name is Faith S. Apayarte. My name is Aisha Nicole B. Gilio.
And we are the Group 5. So this is our introduction for our topic. Your genes carry instructions that tell their cells to produce proteins. And proteins serve a variety of tasks in your body to maintain it healthy. Each gene contains instructions for determining your traits such as eye color, hair color, and height. There are several gene variations for each feature. Genes are DNA segment that contains the instructions for protein synthesis, which is necessary for the structure, function, and regulation of body's tissues and organs. Proteins have diverse functions in our body because it includes catalyzing biochemical reactions and it serves as signaling molecules. And now let's proceed to the first topic of our chapter. We have lesson 7.1, Genetics, a 21st century science. Genetics is an essential branch of science since genetic products are present in our daily lives. In the 21st century, a considerable portion of the food we eat is genetically modified organisms or contains GMO-derived components. According to the statement, genetic products are very common in our daily lives, and genetic technologies and their outcomes are widely utilized and very impactful in very different industries, especially for agriculture, medicine, and biotechnology. When we say GMO, it means genetically modified organisms, and these organisms whose genetic material has been transformed using genetic engineering techniques is introduced decide features, or they are the one who introduce decide features of an organism. And now let's proceed to the next slide. Genetic engineering is seen to have the potential for solving a wide range of human issues, including pollution, energy shortages, food shortages, climate change, expanding illness, and overpopulation. Genetic engineering is recognized or considered as one of the most transformative technologies in our modern era because it holds a potential for addressing a wide range of human issues, starting from agriculture to global health crises to human issues in our society. For example, genetically modified crops is a solution in aiding the soil restoration and provides agricultural sustainability. In terms of food security, genetic engineering provides new methods in preventing hunger and malnutrition. In short, genetic engineering is very diverse and very open to social issues such as from the soil to agriculture to medicine and many more. And now, let's proceed to the next slide. In this slide, as we can see, we have the genetics through the years. We have 1982, 1990, 1993, 1995, 1997, 2003, and 2010. For 1982, we have the recombinant insulin as the first commercial product produced by recombinant DNA technology. The development of recombinant insulin is a big improvement for the industry of biotechnology and medicine. And when we say recombinant insulin using DNA technology, this is a treatment for those persons who have diabetes. Before the development of recombinant insulin, the insulin therapy that is used by the person who have diabetes are extracted from pig and cow's pancreas. But pig and cow pancreas have disadvantages and also limitations because there is a risk of allergic reactions in some patients. And now, let's proceed to the year 1990. This is the first use of gene therapy to treat SCID. When we say SCID, it means severe combined immunodeficiency. SCID, also known as bubble boy disease, it is a rare genetic disorder of severe immune system deficiency that affects an individual to be extremely prone to any infections. Now, let's proceed to the year 1993. We have the Flavor Saver tomato, the first FDA-approved GMO. It was the Colgene Incorporation. It is a biotechnology company that first implemented the Flavor Saver tomato. And when we say Flavor Saver tomato, it is a very innovative technology that addressing the common issue of tomato production. For example, premature softening of tomato and deterioration during shipping and storage. Now, let's proceed to the year 1995. It was the first use of DNA tests in court in O.J. Simpson trial. The use of DNA testing was central to this case, as it was the first time such scientific proof has been used extensively in a criminal trial. Now, let's proceed to the year 1997. In this year, it was when the cloning of Dolly the sheep. The cloning of Dolly the sheep was a very historic event in the industry of biology, as it was the first time a mammal was cloned from adult somatic cell. And last, for our genetic through the years, we have 2003 and 2010. For 2003, it was the completion of Human Genome Project, and for 2010, it was the creation of Cynthia, the first synthetic microbe. Now let's proceed to the 7.2, which is the inheritance of traits. Now, for our introduction, when you hear the word inheritance, what word that comes into your mind? When I hear the word inheritance, the word that comes into my mind is passed down. Why? Because inheritance for me is when an individual bequeaths something from their loved one or parents. Same as in the biology, which is the inheritance of traits, where the traits of the parents will be passed down from their offspring, which making them have the same appearance, traits, behavior, and the physiological processes of the resulting organism. To another introduction, parents pass on features or qualities such as eye color and blood type to their children through their genes. Some health issues and diseases can be handed on genetically. Sometimes one quality takes several different versions. For example, blood type can be AB, or O. Another information of the inheritance of traits, which is the genetics. Genetics is the study of how these qualities are passed down across generations. It means when parents have this kind of trait, the offspring will have this kind of trait. Genetics also studies how environmental and non-genetic variables influence offspring's differences or variation. It means that environment can influence the offspring on their differences and variation. This is the process on how inheritance of traits work. When a diploid cell with homologous chromosomes undergoes meiosis, the alleles split, which is where the genetics of the parents contains, resulting in haploid cells, which is the sperm cell and the egg cell. When a sex cell is fertilized, the alleles present in that cell are handed down to the offspring. Geneticists utilize the possibility or probability that one allele will be passed down onto a sperm or egg cell to foresee genetic combination. For example, when a mother has blue eyes, the genetics of the mother will be passed down to the children. So the children will have its mother's eyes, which is the blue eyes. And because of the homologous chromosomes that carries all the characteristics of the parents that defines the appearance, behavior, and the physiological processes of the resulting organism. And that's how the inheritance of traits works.
So now, let us proceed to 7.3 genotypes control phenotypes. The totally of genetic makeup of an individual is called genotype. A phenotype refers to the observable expression of a particular genotype.
For our 7.4 lesson, we have the Mendel's law of inheritance. Mendel's law of inheritance, this was illustrated by the Augustinian monk named Gregor Mendel. Well, according to Mendel, there exists a pair of genetic material in every organism that segregate during gamete formation.
So, this is monohybrid cross. Monohybrid cross is like predicting the traits of the children when two parents have a different eye color. So let's proceed to Punnett square of monohybrid cross. So it is a tool to guess the chances of certain traits appearing in children, like whether they have a brown or blue eyes. So next, the dihybrid cross. So this is when we're looking at two traits at once, such as eye color and hair color, to see how they're inherited together. So, in summary, monohybrid crosses focus on the inheritance of one trait. Punnett squares help predict the possible outcomes of genetic crosses. And dihybrid processes involve the study of inheritance patterns for two different traits simultaneously.
And these are the contribution of Mendel to the science of genetics. The first one is we have unit factors in pairs. These genetic characters are controlled by the unit factor that exists in pairs in individual organisms. The second is the dominance and recessiveness, in which when two unlike unit factors are responsible for a single trait are present in an individual, one unit factor is dominant in the other, which is said to be recessive. The third is the segregation. In this segregation, during this, the formation of gametes, the paired unit factors segregate randomly so that each gamete receives one or the other with equal likelihood. And lastly is the independent assortment. During gamete formation, segregating pairs of unit factors are assorted independently of each other. And that would be all for the lesson 7.4.
Now, let's proceed to the lesson 7.5, the non-Mendelian patterns of inheritance. Non-Mendelian patterns of inheritance refer to traits that follow different inheritance mechanisms. Non-Mendelian inheritance is classified into 8 types: dicodominance and complete dominance, lethal genes, multiple alleles, polygenic inheritance, sex-linked inheritance, sex-influenced inheritance, and biography. Now, let's first discuss what is codominance. So, codominance, it is a phenomenon in which two alleles and different versions of the same gene are expressed equally in an organisms. So, in addition, codominance occurs when two different allele at a gene locus are both fully expressed in the phenotype of an organism. An example for this will be the human AB blood group system. And now, let's proceed to the incomplete dominance. It is the type of gene interaction in which both alleles of the gene at the same locus are partially expressed, frequently resulting in an intermediate or different phenotype. It is also referred to as partial dominance. So, incomplete dominance is a situation where the dominant allele does not completely mask the effects of recessive allele into an heterozygous condition, leading to an intermediate phenotype. Next, the lethal genes. Lethal genes. It is a gene that under certain conditions can prevent development or kill an organism or its germ cells. It is also known as a lethal factor, lethal mutant, or lethal mutation. So, lethal genes are alleles that cause the death of an organism carrying them. They can be dominant, recessive, or conditional. Next, the multiple alleles. It is a genetic locus allele that exists in more than 2 allelic forms in the population. So basically, multiple allele refers to the existence of more than 2 alleles for a genetic locus within a population. A classic example for this again is the ABO blood system.
So let's now move on to polygenic inheritance. This happens when two or more genes controls a trait which create variations. Examples of polygenic traits are height, eye color, and intelligence quotient or IQ. Is sex-linked inheritance. It is a biological process that involves the transfer of traits or characteristics from parents to offspring. This inheritance of genes or traits comes from sex chromosomes. An example of this is inheritance of color blindness from mother's X chromosome. Sex-influenced inheritance. These are autosomal traits that are affected by sex. If a male has one recessive allele, he will exhibit that trait. However, the female will require two recessive alleles to exhibit the same trait. This is where the dominance of trait is affected by the sex of an individual. An example of this is inheritance of baldness, which is dominant in males. Pleiotropy. It is a type of genetic expression in which a single gene influences several traits. An example of this is beta-globin gene mutation or sickle cell anemia. This can lead to blindness, liver failure, and heart attack.
Proceed to 7.6. Pedigree analysis. So pedigree analysis is a genetic family tree that shows which members of a family are affected by the trait being traced. It also includes standardized symbolized base family history diagram. So here is symbols in a pedigree. Here is male not expressing the trait, female expressing the trait. And same goes to female. And here is the identical twins, paternal twins, offspring, and siblings. And that is a symbol in a pedigree. And then here is the example of inheritance of hemophilia in the descendants of Queen Victoria and England. So here you can see that Albert passed through to Edward, then Edward passed through George, then George VI. And that's it. Well, Albert also passed to Alfred, Arthur, then so on and so forth. So here is, this is the normal male, and then this kind of color is the hemophilic male, and that's it.
So now let us proceed to topic lesson 7. So producing genetically modified organisms. Genetic modification or GM is technology that inserts DNA into an organism's genome. To create a GM plant, new DNA is introduced into plant cells. Usually, the cells are grown in tissue culture until they develop into plants. The seeds produced by these plants will carry the DNA. So, an organism genome is the complete set of genetic information of an organism. It all contains the instruction for creating and maintaining life. The next one is genetic engineering. Genetic engineering allows genes from one organism to transfer it into the DNA of another organism. So genetic engineering, it is a scientific technology to copy or change or replace the genetic structure. Now we have recombinant DNA technology. It is a technique of combining two DNA sequences from different sources. So recombinant DNA technology, it is a technique of combining two DNA from different sources to create new genes or new functions. The next one is we have genetically modified plants. Plants with pest resistance. It makes crops resistant to NCPs without using chemical pesticides, which is beneficial to the environment. So the next one we have
plants with herbicide resistance. Glyphosate is a powerful herbicide which kills actively growing plants. It also stops the crops from releasing essential aromatic amino acid which they need for growth. The next one we have plants with lower nutritional values. The most promising value of genetically modified crops is in the production of genetically engineered plants with desirable traits and with great benefits to the consumers. So, genetically modified food consumption and its potential risk. So the first one is it could pose potential harm to other organisms. And the next one is eliciting resistance. And the last one is disrupting or affecting genetic flow. So the next one we have genetically modified animals, transgenic animals for manufacturing a product. So gene farming makes use of transgenic animals to produce pharmaceutical products for therapeutic and diagnostic. So below, animals are used to make human proteins that have medical value. So the last one we have cloning transgenic animals for various purposes. Animal cloning is a difficult process with a low success rate, and most clones are dying early. So we all know that animal cloning is a tough or difficult process with a low success because it causes the animal suffering.